I hope every one of you are fine and well, and also you guys are safe. I welcome you all to Gurmantra Academy, and today it's our cell biology lecture. So we're going to see some more in detail about the cell signaling class. Okay. So today we are going to learn few pathways in detail. So one such important pathway is your cyclic AMP pathway. So what does cyclic AMP means? It means cyclic adenine. Okay, so A is basically adenosine. Okay, it's adenine. You know, adenine, guanine, cytosine, thymine. So that A is only incorporated here. So it's basically adenine group which is added with one phosphate. So adenosine monophosphate. Okay, adenosine monophosphate is your cyclic AMP molecule. Okay, so why are we studying about this? What is this cyclic AMP? It is a secondary messenger. Okay, it's a secondary messenger and it is important in several biological processes and even in pathways. Okay, so it is actually derived from ATP. You know, adenosine triphosphate. It will lose two phosphate groups. Okay, two phosphate groups, that is nothing but your PPI. It will lose and you will get AMP. Okay, so that is your AMP. Okay, and now this AMP in its cyclic form, it's called as cyclic AMP. Okay, okay, this cyclic AMP can also be called as adenalic acid. Okay, it can also be called as adenalic acid. So cyclic AMP is synthesized from ATP. It is basically synthesized from ATP. Cyclic AMP is synthesized from ATP. We just saw now. Okay, so what happens is ATP in the presence of the enzyme adenylate cyclase. Adenylate cyclase. Okay, what it will do is it will convert ATP to cyclic AMP. Okay, it will convert ATP to cyclic AMP, and now this will be useful in several reactions in your G protein coupled receptors. Okay, so can you see in this picture you have ATP in the presence of the enzyme adenylyl cyclase, you are getting cyclic AMP. Okay, so G protein exists either in an active or an inactive state. Okay, if it is bound to GDP, which means it is inactive in nature, and if it is bound to GTP, it means it is active in nature. Okay, it is active in nature. So in the active state, what happens is the G protein will be bound to GTP. This should be very clear. Also, you should know that G protein, they have ATPase activities. Okay, they have ATPase like activities. Since here GTP is involved, we call it as GTPase activity, which means what? It can convert GDP to GTP or from GTP to GDP. So the hydrolysis and phosphorylation dephosphorylation reaction is taken care by the enzyme GTPase. Okay, so this is what happens in a cyclic manner. So what happens? Ligand will bind to the receptor. This is the first step. Okay, now what will happen? G protein will be activated. G protein will be activated, and how will it be activated? Because it is attached to GTP. Okay, it is attached to GTP. Now what will happen is, since this GTP, okay, this G protein is activated, now what will happen is your G protein will get split up. G alpha subunit will be split up from beta and gamma subunit. Okay, G alpha GTP. G alpha GTP. Now to this G alpha GTP, you have the enzyme adenylate cyclase. Okay, adenylate cyclase enzyme will be used to convert the adenylate cyclase enzyme will be used to convert ATP to cyclic AMP. Now this is the secondary messenger. Now this secondary messenger will be further used on for the reactions. Okay, so different G proteins has a function that they can either stimulate the pathway or they can inhibit the pathway, based on which it will have the effect on cyclic AMP. For example, if this G stimulatory, so what it will do, it will activate the adenylate cyclase. So your cyclic AMP concentration will go high. And you know another G protein, G inhibitory, if this is activated, okay, if this is activated, what happens is it will inhibit the adenylate cyclase. Then cyclic AMP levels will drop. Okay, its concentration will drop. So based on the different G proteins, it can either act like a stimulatory protein or an inhibitory protein. Okay, so can you see in this picture, you have the hormone, which is nothing but your ligand. Okay, now that is coming and binding to the receptor. Now receptor is activated. Okay, now receptor is activated. When the ligand binds to the receptor, it becomes active in nature. When it becomes active in nature, your G protein, okay, ligand binds to the receptor, then what happens? Receptor is activated. Okay, receptor is activated. Now next what happens is, there will be conformational change. Conformational change of receptor. Conformational change of receptor, which leads to activation of G protein. Which leads to activation of G protein. What is that it means by activation of G protein? It means that G protein is attached to GTP. So this complex is called as active protein. Now when G protein is attached to GTP, what will happen? It will have the alpha subunit. Alpha subunit will get separated from the beta and gamma subunit. Okay. Now beta and gamma subunit will be present in the plasma membrane. This will be present in the plasma membrane. And this G alpha here, which is along with the GTP, G alpha GTP, along with the GTP, will go and carry on its function. What is its function? The first function is it will activate adenylate cyclase. Okay, it will activate adenylate cyclase, which is an enzyme, which is a very important enzyme. What this enzyme does is, this enzyme will convert ATP to cyclic AMP. Okay, ATP to cyclic AMP. Now, what is this cyclic AMP? It is a secondary messenger. It's a secondary messenger. It has important functions in the processes. Okay, I think you can understand the process up to this, right? Ligand will come and bind to the receptor will get activated, there will be conformational change of the receptor that will cause the activation of G protein. Again, when G protein is active, it will be bound to your GTP, right? Now when it is active, the alpha subunit and beta gamma subunit will be separated from each other. The beta and gamma subunit will be present on the plasma membrane itself, the cytosolic side. The G alpha will actually separate from them and go inside this cell. Okay, it will be going inside the cytosolic region. So what happens is, now this G alpha GTP will activate an enzyme called as adenylate cyclase. Now what this adenylate cyclase does is, it will convert ATP to cyclic AMP. Okay, what is your cyclic AMP? Cyclic AMP is a secondary messenger. Okay, cyclic AMP is a secondary messenger. Now this will activate several protein kinases. This will activate several protein kinases and it will lead to metabolic events. Okay, it will lead to metabolic events. So this is the process of your G protein coupled receptors. Okay, so this is the process of your G protein coupled receptor. Do you understand the process? Do you understand it? I hope you understand it. Okay. So when the work is done, I mean when all of the process is over, there should be deactivation of the receptor. It cannot be active all the time. That will cause a problem again. It has to be restored to its own state. So then what will happen? What will happen is, your G alpha GTP, you have this G alpha GTP, right? G alpha GTP. we know that the G protein has GTPase activities. So what it will do is it will convert G alpha GTP to G alpha GDP. Now this is an inactive protein. This is an inactive protein. Now this will go and attach itself with beta and gamma subunit. Again, now this will cause the desensitization of the receptor. The receptor will be desensitized. Desensitization of the receptor, and the ligand will go outside. Now it will be normal again. It will be in a state where another ligand can come and bind to it. Okay, so this is your G protein coupled receptor cyclic AMP pathway. Cyclic AMP pathway. Okay. Okay, so what are the hormones that will transduce the signal, that will act like a ligand? That will act like a ligand for your cyclic AMP pathway. So you have epinephrine, norepinephrine, glucagon, adrenocorticotropic hormone. You have vasopressin, luteinizing hormone, thyroid stimulating hormone. So these are some of the ligands which can activate your cyclic AMP. Okay. Now whatever I have taught you, I have made it into a diagram so that you can easily understand it. What you can do is take a screenshot of this particular slide and you can study them. Okay, you can use it for studying because I've made it everything simple and it's like a handwritten notes. So you know that G protein coupled receptor is a serpentine receptor, which means it will span the membrane 7 times. 7 alpha helical structures will be present. Okay, so it will also have a ligand binding region, it will have a transmembrane region and a cytosolic region to which G protein will be attached. Okay, you can see here ligand is binding to the receptor, which is a serpentine receptor. Okay, now that causes conformational change in the receptor and it activates the G protein. When G protein is activated, which means G alpha GTP, okay, G alpha GTP, and for this activation you need guanine nucleotide exchange factors. Okay, guanine nucleotide exchange factors that will only cause G alpha to be attached to GTP. When this is done, the alpha subunit will get separated from beta and gamma subunit. Beta and gamma subunit will be present on the plasma membrane itself and your alpha subunit. Now this subunit is the active part of the G protein. It will go and activate. It will go and activate the enzyme adenylate cyclase. What adenylate cyclase does is whatever ATP is present inside the membrane, it will convert ATP to cyclic AMP. Now what is cyclic AMP? It is your secondary messenger. Okay, it will further do its function. Okay, it is helpful in the transduction process. Transduction of signal. Okay, so secondary messenger. What this secondary messenger does is it will activate protein kinase A. pKa is nothing but protein kinase A. Protein kinase A. This will activate protein kinase A. Okay, so what happens is you will have regulatory subunit and catalytic subunit like this in protein kinase A. Okay, your cyclic AMP will come and bind to regulatory subunit. Okay, it will come and bind to regulatory subunit. So what will happen to that? What will happen to that is when cyclic AMP is bound to the regulatory subunit and catalytic subunit will detach from each other. Okay, it will detach from each other. So this causes activation of the catalytic subunit. Okay, activation of the catalytic subunit. This is what is important in the function. Okay, activation of catalytic subunit. Now what this activated protein kinase A, which contains only the catalytic subunit, do is it will phosphorylate several proteins. Why does it phosphorylate? Because it is protein kinase. The function of kinase itself is to phosphorylate, right? So it will phosphorylate several proteins. Okay, and eventually what is the result? It will regulate the gene expression, it will help glucogen phosphorylase, then it will help in the muscle contraction, increased heart rates, like that. Also now this activated protein kinase A, it can enter inside the nucleus. It can either, okay, these are the two pathways, one in which it will act like a kinase and phosphorylate several reactions and proteins. Second, it can itself enter inside the nucleus and it can transcribe CREB. Okay, it can transcribe CREB, which is a part of gene expression. Okay, so regulation of cyclic AMP can be done by your adenylate cyclase and your phosphodiesterase. Okay, phosphodiesterase. So what is the function of adenylate cyclase? If you are inhibiting adenylate cyclase, what will happen? ATP to cyclic AMP process will not take place at all. That is one kind of regulation. Second, since you have lot and lots of cyclic AMP, which is a secondary messenger. Now, if I convert this into 5-AMP, which is not a secondary messenger, not a messenger. Okay, it is not a secondary messenger, this 5-AMP. Now, this cannot activate any protein or cause gene expression. Okay, so this cannot do that. So, phosphodiesterase can help in this function. So, this is how regulation of cyclic AMP pathway is done. Okay, so this is your summary slide. You can take this as a revision box. Whatever I have explained you is given in a written format. Okay, the hormone receptor will bind. Okay, that will activate G protein. How? G alpha GTP. Okay, now this will separate from beta and gamma subunits. Now this G alpha GTP will activate the adenylate cyclase. What this adenylate cyclase will do is it will convert ATP to cyclic AMP, which is the secondary messenger, that will activate your protein kinase A. Okay, and in this what happens, the regulatory subunit will be cut off from the catalytic subunit. Okay, catalytic subunit is the active part of protein kinase A. Now this will cause several metabolic reaction to happen. Okay, now down regulation. So the hormone has to unbind. GTP will be converted to GDP using the gap. Okay, adenylate cyclase should be inactivated and phosphodiesterase enzyme will convert your cyclic AMP to 5-AMP, which is not a secondary messenger. Okay, do you understand the process? So here is an example. Okay, some heterogenic receptors in the plasma membrane, they respond to the epinephrine. Okay, they respond to the epinephrine. And how do they do that? So you have a liver, inside that you have a liver cell. In the plasma membrane of the liver cell, you have G proteins. Now the epinephrine is coming and binding to the G protein, activation of the receptor, activation of G protein. So what happens? G alpha GTP will be activated, that will activate adenylate cyclase, that will convert ATP to cyclic AMP, then that will activate protein kinase A, and further several glucose metabolisms will happen. Okay, this is how the amplification of signal happens. Okay, amplification is very important to understand, because it is not like when one particular ligand binds, only one signal is sent. No, it is amplified in between. So you can see one norepinephrine is binding to the receptor, but that can activate several G protein. Okay, now that will activate the adenylate cyclase. So adenylate cyclase can convert several ATP to cyclic AMP, a lot. Now this cyclic AMP will, what it will do, it will activate your protein kinase A. Now this protein kinase A can phosphorylate several proteins. So this is how your amplification will take place. Okay, so the molecules that can activate the cyclic AMP pathway, this is very important slide. Okay, potential MCQ questions. So cholera toxin, caffeine, pertussis, all of this can activate the cyclic AMP pathway. And what are some of the molecules that inhibit the cyclic AMP pathway? You have phosphodiesterase enzyme and GI alpha, G inhibitory. Okay. So next pathway we are going to study about is IP3 DAG pathway. Okay. In IP3 DAG pathway, if you see, it is also similar to that, your G protein coupled receptor only, but here some of the enzymes are very different in nature. So you can see here your ligand is binding to the receptor. Okay, that causes activation of the G protein coupled receptor. Now what this will do is, this will activate an enzyme called as phosphorylation phospholipase C. Phospholipase C. What this enzyme does is it will break down PIP2 into IP3 and DAG. You have a homework here. Now you have to find out what is the full form of PIP2, PIP3, IP3, okay, and DAG. DAG is easy, I can tell you, but I know this, but I want you to find out because these full forms are also asked as MCQ. So this is your homework question. Okay, so now what happens is PIP2 is converted into IP3 and DAG. DAG will be present attached to the membrane to its cytosolic region, but IP3 can go to the smooth endoplasmic reticulum, which will have the channels. What channels? Calcium ion channels. It will open the calcium ion channels, and the concentration of calcium inside the cytosol will increase. Okay, and that can also helpful in the several pathways. Now, what are the ligands that can be used in IP3 DAG pathway? It can be a neurotransmitter, a hormone, a growth factor, angiotensin 2. These are MCQs. Again, potential MCQs. Okay, can you see here? I will just revise it for you. Okay, this is your revision box. Can you see here the ligand is binding to the receptor, which is a G protein coupled receptor. That is why it is serpentine in nature. 7th alpha helix is present. Now that will activate your G protein. Now G protein is activated means what happens? Here the G protein is GQ. Okay, here the G protein is GQ. Now what will happen? The alpha subunit will get separated from beta and gamma subunit because of GTP. Now this GQ alpha GTP, what this will do? This will activate the phospholipase C enzyme. It is an enzyme. Okay, it will activate this enzyme. What this enzyme will do is it will convert PIP2 into IP3 plus DAG. Okay, so this conversion actually takes place. DAG will be present on the plasma membrane itself. IP3 will go into the endoplasmic reticulum, especially smooth endoplasmic reticulum. It will open the calcium ion channels. Calcium will come out of the cell and its concentration will be increased inside the cell. Now, this calcium along with the DAG, okay, calcium along with the DAG, it can activate protein kinase C. Okay, protein kinase C is again an enzyme. Now this can phosphorylate several proteins. So this is your IP3 DAG pathway, which is actually a G protein coupled receptor. So let's see some of the functions. Let's see some of the functions and applications of this. The first one is that this IP3, it will cause calcium ions increase inside the cytosol. Second, because calcium ions is increasing, it causes smooth muscle contraction along with the calmodulin, like that. Next, IP3 in the brain. In the brain, it helps in the synaptic plasticity. Synaptic plasticity means indirectly strong memory formation. Okay, again, in sea urchin to prevent polyspermy, slow block reactions are used. Okay, slow block polyspermy reactions is used in sea urchin in the fertilization process of an egg and a sperm. If you want to read more about it, you can actually read because it's a very interesting topic. Okay, but it's just an example. They might not ask you this for your exam. It's just an example. Okay, so next thing we are going to learn about is cholera toxin, which also is a function of G protein coupled receptor. So what is cholera? It causes diarrhea, and that is because of contaminated food and water by the microorganism Vibrio cholerae. It's a gram-negative bacteria. Okay, so cholera toxin can be written as CTX या फिर choleragen enterotoxin. Okay, what it is made up of? It is made up of two A subunit and 5 beta subunit. A subunit will contain A1 and A2. The 5 beta subunit is same, 5 beta subunit. So A is the subunit which has the enzymatic activity. B is the subunit which is binding to your receptor, and it is denoted as AB5. Important MCQ question. So mode of action is that ligand is your cholera toxin. Receptor, the G protein coupled receptor on which this cholera toxin binds is called as ganglioside receptor. And where is it present? It is present in the intestinal epithelial cells. Okay, what happens? You have to understand a process called as ADP ribosylation. ADP ribosylation means to ADP. Okay, addition of ADP ribose to a protein, that is called as ADP ribosylation. ADP ribose is a structure. Okay, now this will go and attach itself with a protein. Now that is called as ADP ribosylation, and it is a process which is happening in your post-translational modification. Okay, it is involved in DNA repair, then gene regulation and apoptosis. So can you see here? You have your sugar, a base, that is adenine base, okay, 2-phosphate and a ribose group which is replaced. Okay, here ribose group is attached in place of a phosphate group. One phosphate group has been removed and a ribose group is attached. So now it is ADP because only 2-phosphate is present plus ribose. That is why it is called as ADP ribosylation. It will be done to a protein. Okay, so let's see the mechanism of action of your cholera toxin. Okay, mechanism of action. Can you see the cholera toxin which contains A beta 5 subunits? Beta 5 is the subunit which will bind to your ganglioside receptor. Can you see it is binding to the receptor? Okay, endocytosis will take place. Endocytosis means along with the membrane a blob will be formed and this will be entering inside along with your toxin AB5. Now this will go inside the endoplasmic reticulum where only A subunit will exit from the vesicle. Beta subunit B5 will be present inside the vesicle. Only A1 subunit will come out and it will cause G protein. Okay, it will cause G protein to be ADP ribosylated, which means it will add ADP ribosylation to your G protein. So what will happen is your G protein will be not having GTPase activity. Okay, G protein will not have GTPase activity. Why is GTPase required? GTPase will inhibit the G protein function. Now if you are itself inhibiting this GTPase, now this will be always active. So if G protein is always active, then your adenylate cyclase will always be active. Cyclic AMP level will always be high. Protein kinase A is always active. So this will phosphorylate your CFTR, cystic fibrosis transmembrane receptor. It will activate it and that will cause diarrhea. Just because your G protein is not returning to its original state, it is causing full-time activation of the pathway. That is the reason for watery diarrhea. Okay, that is what is your cholera toxin doing. Next is your pertussis toxin. Pertussis toxin is produced by Bordetella pertussis. It causes whooping cough and here the process involved is GI alpha. Here the G protein is GI. And this results in the cyclic AMP which will cause loss of fluids and electrolytes. Okay. When there is no pertussis toxin present, this is case 1 when there is no pertussis toxin present. So what will happen? GI will be there. Okay, GI will be there. Now what this GI does is, what is the function of G inhibitory? G inhibitory will inhibit the adenylate cyclase. Okay, it will be inhibit the adenylate cyclase. So cyclic AMP levels will always be low. Okay, but what happens when pertussis toxin is present? Now when ligand, that is the pertussis toxin, is binding to the receptor, now this will cause ADP ribosylation of GI. Okay, ADP ribosylation. Can you see here? ADP ribosylation of GI. Now GI is inactive. Okay, so what is the function of GI? When GI is active, it will inhibit the adenylate cyclase. If I am inhibiting GI itself, then what will happen? This function will not be there and your adenylate cyclase is active always. So more cyclic AMP will be produced and that will lead to whooping cough. So this is the function of your pertussis toxin. And next pathway we are going to learn about is GPCR in vision, G protein coupled receptors in vision. So they are basically two types of photoreceptors. One is your rods, the other one is your cones. Okay, so rods are involved in the night vision. Okay, rods are involved in night vision because only a very few photons itself is enough to activate these cells. Okay, cones is for the daylight because lots and lots of photons are required for activation of your cone cells. So vision in the eye is also dependent upon your G protein coupled receptor, but here it is an ion channel. Okay, here it is an ion channel. Let's learn few terms. First is rhodopsin. Rhodopsin is nothing but your G protein. That is only, that is your receptor. Transmembrane protein receptor, which is a chromophore in nature. It has 11 cis retinal attached to the opsin. This is the structure basically. Next is your transducin, that is GT. Now this is your G protein. Okay. Next is phosphodiesterase enzyme. This, what is that? What is the function of PDE? We've already studied. It will convert cyclic GMP to 5-GMP. So phototransduction in the rod cells. Okay, first case, in the presence of light, what happens? When the light is present, no neurotransmitter is required basically. Rod cells, okay, so rod cells, when light falls on the rod cells, all of the rhodopsin protein, which is an G protein, it is getting activated. What it will do is it will convert all the cis retinal to trans, all trans, 11 cis retinal to all trans. So this will activate your transducin, that is G protein, GT, transducin will be activated. Now what this will do is this will activate the phosphodiesterase enzyme. This will activate the phosphodiesterase enzyme. What this enzyme does is it will convert your cyclic GMP into 5-GMP. When 5-GMP is there, so what is 5-GMP? It is not a secondary messenger, which means all of the cyclic GMP is converted into 5-GMP, its level will become low, right? Because all of it is getting converted. So cyclic GMP level is low. Why is cyclic GMP important here? Because that is a secondary messenger. So this will affect the sodium channels. Sodium channels will be closed. There will be no entry of sodium ions inside the cell. That will lead to hyperpolarization. Hyperpolarization means your membrane potential is sinking to negative, to -71, to -17 millivolt. Okay, so this causes decrease in the synaptic transmissions. Okay. Next, at night neurotransmitters are basically required for you to see the objects. Okay, so at night what happens is rhodopsin, okay, the rhodopsin opsin protein is there. Okay, now this is not converted to from it is not converted from 11 cis retinal to all trans. So that process is not taking place. So what happens? Your transducin will not be activated. Your cyclic GMP phosphodiesterase will not be activated. So what happens? Sodium ion channels will be always open and it will lead to depolarization. Depolarization is the first step in your action potential and it will help in synaptic transmission. The synapse to open, transmitters to be released. Okay, only then you'll be able to see the objects in dark. Can you see here? This is your opposite protein and in between you have 11 cis retinal. When light stimulation of the rhodopsin leads to the activation of the G protein. When light is falling onto the rhodopsin, G protein is activated, that is GT alpha GTP. Now what this will do? It will activate your phosphodiesterase enzyme. You know the function of phosphodiesterase is it will convert cyclic GMP to 5-GMP. Now since there is no cyclic GMP present, your your sodium channels will be closed. When sodium channels is closed, it will lead to hyperpolarization. Hyperpolarization means the membrane potential is negative in nature. So what happens because of this? Neurotransmitters will not be released. Okay, because neurotransmitters are required when you want to see in dark. But when light is present, neurotransmitters is not required. Okay. Okay, so can you see here when light is falling on a receptor, photoreceptor, your rhodopsin is activated. It will activate transducin, which is your G protein. That will again activate phosphodiesterase enzyme, and your cyclic GMP is converted to GMP, 5-GMP. The channel will be closed. Sodium ions cannot enter inside the channel. Okay, so this is what is given here. Okay, now amplification. When we talk about amplification, when activation of the receptor take place, one photon will activate one rhodopsin, one receptor. But one rhodopsin can activate hundreds of G protein, that is transducin. Again, one transducin can activate hundreds of phosphodiesterase enzyme. Then one phosphodiesterase enzyme can again convert, it will convert several of your cyclic GMP to GMPs. So can you see there is always amplification happening. Okay. Okay, so these are the questions for your homework, which is completely about your G protein coupled receptors. Okay, this is a homework, very simple questions. If you have listened to my class, you can easily answer them. Take a screenshot of these questions. You can answer these questions as your homework. Okay, I hope you liked the class today about the G protein coupled receptors and the various pathways associated with it. Okay, you can like, share and subscribe to our YouTube channel Gurumantra Shikshaka Granth. You can join our Telegram Chandra Gurumantra Academy. Okay, if you type in Telegram Gurumantra Academy, you can join the channel. Okay, next is you can join our Facebook channel and you can visit our website as well. If you have any doubts, you can call us or mail us, whatever it is. We are here to help you all the time. So thank you so much guys. Keep preparing for your exams very well. All the best.